And welcome to the second day of the GIAN course. I again welcome Professor Vasileios. Since it is Sunday, it's off day.
No,
But still, since the course schedule is such that we are having it from 9 to 30 Sunday there, so we'll be bothering you, right? Right, sir. All yours.
Thank you so
So
much.
we'll be having two lectures, then we'll be having a break for tea, lunch, and then we'll have a practical session in the lab where demonstration will be organized. We will be demonstrating certain the methods and the methodology, how to go for priming, chemical, biological, and the students, they have already prepared certain things for you. And the overall background will be provided by Professor Vasileios.
Perfect.
Thank you.
Perfect.
Thank you.
Thank you so much, Professor Sharma. Good. So, thank you for being back here, even on a Sunday. Hopefully, it will not be too tiring today. And today, basically, the approach will be to follow, based on the schedule that was given. I feel it is smoother to actually start off with a general overview in plant stress principles. And then I will follow by mentioning key plant responses to stress. So this should be the focus of today's approach, and then moving on, we will switch to the involvement of signaling molecules, which is a very important issue because a lot of these molecules can effectively act as priming agents themselves. And then later on in this series of lectures, we will actually focus on what I think is the most interesting part, which is the case studies. So basically, really going deeper into the chemical priming approaches, the various biological priming approaches, as well as the nanomaterial-based priming approaches. So kind of going through case study scenarios. Okay? As I said yesterday, it is a great pleasure if you interrupt and ask questions. I prefer it if there is interaction during this procedure. And at the end of the day, it's always welcoming to have a very vibrant discussion, brainstorming session, so that we all start thinking about this concept in a more active manner. Okay? So this is just as I mentioned before. So basically, this photograph is not randomly taken. This photograph was taken from The Independent, which is like a UK newspaper that was headlined quite dramatically some time ago, "The Century of Drought." It was kind of like a call to arms, trying to warn people about the consequences of climate change. As I mentioned yesterday, climate change is upon us. It is severely affecting productivity. As I said, close to 97% of global rural land area is somehow affected by some sort of stressor. And actually, pictures like this are not really that uncommon nowadays. So, our focus is not just on the stressors themselves, of course, but it is very important to consider that we have to understand the challenges that plants must actually face in their quest for survival and adaptation, especially considering my comment yesterday about their sessile, their sessile nature, that really takes away some of the key weapons that mammalian systems have for defense, such as the run and flee approach. So there is great amount of tools that are actually in our hands to actually facilitate this approach. So we can actually start from a microscopic level, at a cellular level, organellar even level, to then going in a more broader, morphological as well as agronomic approach. Ending up always in the final observation, which is always, of course, the phenotypic response, which is the most important part of the stress story. So hopefully we will actually start slowly going through the various key points about the responses of these plants. So stress itself, as a definition, it actually encompasses the adverse effects that non-living environmental factors exert on plant growth and development. Of course, there is also the very strong notion of biotic stressors, which is also a major player in the field. But the biggest percentage of agricultural losses is accounted for by abiotic stressors, and not as much as through the biotic ones. These factors include, of course, but they're not just limited to these, extreme temperatures. So depending on the global positioning, you can have heat waves, you can have cold stress, freezing stress, and so on. Of course, water scarcity is a major issue. I assume that in India this is also an issue of importance. Of course, in Cyprus and in the Mediterranean, it is a huge problem. We have variations in light exposure, but also other stressors that I will mention a bit later on, because the list is quite long. It is important, though, to establish a solid definition, because this actually allows us to actually try to look for strategies to actually confront these stressors. And of course, this is a graphic representation I took from Salisbury and Ross, which is a very nice textbook dealing with plant responses to environmental challenges. And this is a very standardized graph that shows basically the relation between the factor level of the environmental Q, effectively represented as the dose, along with the relative plant response. Let me get my pointer. Like yesterday, I think it's useful to point to points of interest. Pointer. Whoop. You have a pointer?
Yes.
Okay. Maybe I forgot it in my room. Sorry. Thank you so much. Perfect. Good. So this is a basic standard representation across the factors of relative plant response and the dose, so representing the factor level. So if you, for example, you consider elements, like nutrition elements, for example, in lower doses, this represents the essential factoring of this element. Under specific concentrations, of course, we're talking about deficiencies, like in the case of minerals, that are also detrimental for plant growth. Of course, where at some point with increased doses, we reach a point of optimal concentrations that actually would represent the tolerance phase of the response of the plant to an external environmental cue. This, of course, is upon levels of saturation when we reach the actual top threshold. But after a specific point of increased dosage, of increased concentration, of course, this then switches to a toxicity zone, thus leading to inhibitory effects in plant growth and plant development and promotion. So further on, of course, this can ultimately result to lethal status, to lethal toxicity, thus leading to the death of the organism. So in addition to the concepts mentioned before, as I said, also there is quite variable terminology that sometimes is quite confusing. So it is important to actually clarify key terms used in the field. So of course, in terms of, for example, responses of plants to water scarcity, first of all, plants are always ideally looking for an escape strategy where basically this is represented by relative phenology of the plant, such as in the case of annual plant extremophytes. And with this case, you actually completely evade the danger of the specific stressor. But of course, upon exposure to stress, then you can have the concepts of either avoidance or tolerance to the stressor. So effectively, this would mean that in the case of avoidance, this is usually achieved through a status of homeostasis and midday leaf water potential. So effectively, common approaches to reach status of avoidance would include water savers, basically strategies of stomatal regulation and water storage, such as in coniferous forest species, and also in the case of water spenders, of species that are heavy in spending water, such as prosopis species that have very deep root systems, thus reaching water reservoirs. The interesting part of the story of stress physiologists is, of course, when we're reaching points of tolerance. And for this, of course, this happens usually in cases where we have a drop in midday leaf water potential. So this can, examples of such cases include xylem resistance to cases of cavitations, as it's in case of calitris plants, but also cases of turgor loss pressure, such as in acacia forest species. So in case of the environmental origins, abiotic stressors encompass a spectrum, a broad spectrum of environmental challenges that, as I mentioned before, include, in addition to temperature and water availability, it really includes soil composition, it includes chemical composition status in the rhizosphere, but of course atmospheric conditions, because you can also have the effects of environmental pollutants. In India, I recently read an article about heavy pollution levels in Delhi, for example, with some very dramatic photography that was included. So this is unfortunately a reality, and actually all sorts of stressors, as well as, of course, high ozone levels, these do represent a threat to the plants, thus exerting a very profound effect on plant life. So the end result of these environmental cues can have many, many different means of impact. Effectively, the most common ones that are mainly addressed are the disruption of very fundamental physiological and biochemical processes in plants. This includes a very broad range of metabolic activities and pathways. Of course, as you're well aware, elevated temperatures can lead to the denaturation of proteins, while at the same time water scarcity, which is very often related with elevated temperature, hampers photosynthesis, but also nutrient absorption from the rhizosphere. And you have to always remember, as I mentioned yesterday, that in real life agricultural scenario, you very rarely, if not never, face single stressors. It is most likely that you will usually have a combination of stressors, both of abiotic as well as biotic origin. For example, heavy waterfall, rainfall, is usually correlated with severe fungal infection due to the high humidity that actually promotes the presence of these pathogens. And even in this case, you can have primary infections as well as secondary infections that are actually occurring within the plant tissue. So it is a very complex situation that is really important to be fundamentally understood so as to try to develop means of amelioration. Of course, you also have the situation of soil salinity that I think also affects regions in India. This has a major effect on osmotic balance. You can have hyperosmotic shock that is occurring, of course, both of ionic nature, such as in the case of salt stress, but you can also have hyperosmotic shock that is non-ionic, which is more correlated with the water scarcity issue. At the end of the day, the common denominator is that this affects the plant metabolism, and of course, it's important, as I said, to understand all of these disruptions at a cellular level in order to devise the appropriate mitigation strategies. So, plants have actually, in an evolutionary manner, evolved several survival strategies. And as I always tell my students back home in Cyprus, their sessility, the fact that they're stuck to the ground, has forced them evolutionarily to actually become very complex in a biochemical and molecular level. As I said yesterday, defense-related gene families are actually very extended, represented by many, many different isoforms. I mentioned the example of the ABC transporters, the example of sugar carriers, the list is infinite. And in the case of Arabidopsis, for example, the genome of Arabidopsis actually includes close to 3,000 membrane transporters, which shows the complexity of this situation out of the 22,000 genes in the genome. So this is why plants were forced evolutionarily to involve these intricate mechanisms to cope with stress, and this of course includes a whole battery of different responses, which includes altered gene expression, so transcription or regulation of the defense responses, synthesis of protective proteins that are universally facing stressors in a horizontal level, as well as the accumulation of compatible solutes. Main key examples of the compatible solutes, who would like to mention a couple? What would you consider key compatible solutes that are mainly linked with homeostasis in osmostability in the cell? I'm sure you're aware of them. Excuse me? Yes, basically ions, can actually be. Calcium is also very important for the signaling effect through the binding with calmodulin as well, but it's definitely a possibility. I was mainly referring to key osmolytes such as proline, for example. Exactly. You have sugars, polyols, many different metabolites that are actually key in the regulation to achieve osmotic stability. But of course, as signal molecules, you can also have the involvement of, for example, calcium ions. So by understanding all of these adaptive responses, us as agriculturists as well as generally researchers can then use these responses to develop the varieties that are resilient to these responses. And this is a diagrammatic representation that actually I think is a very nice roadmap of what is happening from A to Z. Yes. Okay, which is I usually like having graphical representations to my students, because always in a visual manner it's easier to follow the procedure rather than just listening to a person describe it in detail. So let's go through it in detail together. Of course, as I said before, the plants are constantly being submitted to a number of stressors that actually can result in either osmotic stress, oxidative stress, which is generally, depending on the stressor, can have a primary and secondary effect in the plant. So this, of course, as a result, leads to severe damage in the cell, depending on the dose factor, as mentioned before in the relevant diagram. So this, in turn, leads to the disruption of osmotic and ionic homeostasis, the damage as well of functional and structural proteins, as well as membranes. Through the stress, we have the overproduction of the reactive oxygen species and the reactive nitrogen species. So this effectively leads to programmed cell death, apoptotic responses in the cell, that actually also affects fragmentation of nucleic acids. You have the denaturation of pigments, such as chlorophylls in green tissues, and so on and so on. So it is important, first of all, for the plant to be able to sense this damage, this danger. So we have first the concept of the signal sensing in response to the exposure to the stressor, the perception of this signal, and then, of course, the transduction, because it is very important to have the systemic signaling in more distal tissues. Of course, responses can be both local and systemic, but of course, it is very important to have this transduction that is usually achieved through the vascular tissues. Okay? So, in this case, we have a great battery, again, of means to sense and perceive this signal. So we can have the concept of osmosensors, such as potassium transporters and so on. We have, of course, phospholipid cleaving enzymes, such as phospholipase D. We have secondary messengers, such as the calcium ions that was mentioned before quite correctly, as well as, of course, reactive species. We have the involvement of MAP kinases, calcium sensors, such as the component SOS3 in the salt overly sensitive pathway that was mentioned briefly yesterday, calcium-dependent protein kinases, so effectively CDPKs, and so on. So again, it is a very complex situation using many different receivers of the signal. This, in turn, will lead to the transcriptional control of the response, which is fundamental on the plant's effort to actually address the stressors, and this is mainly manifested through the regulation of key transcription factors that are stress specific, although some of these transcription factors do appear to have a universal role in the response. Major transcription factors that are known to be involved in the transcriptional regulation of defense-related transcripts include CBFs that are quite heavily linked with cold stress responses. We have the DREBs, the drought responsive element, transcription factors that are linked mainly with drought stress, but also with other forms of abiotic stress, HSFs, B-zip transcription factors, MYCs, MYBs, and so on. Okay? As a result, this then will activate the stress-responsive mechanisms. So this can include, through gene activation, components of detoxification. Again, briefly yesterday in my background talk of the work we do in my group, we mentioned the necessity of activation of number of antioxidants, enzymatic antioxidants in the cell. That includes superoxide dismutases that actually dismutate superoxide radicals to hydrogen peroxide, peroxidases, catalases, glutathione reductases, and so on. Okay? We have, of course, the element of osmotic protection that is especially key for stressors that actually disrupt osmotic homeostasis, such as salt and drought stress. And in this case, as I said, it is important to actually activate biosynthetic enzymes leading to these compatible solids. So effectively, for example, the upregulation of P5CS and P5CR that are key enzymes involved in the biosynthesis of proline. Glycine betaine, which is actually another wonderful example, and which is commonly used, now that I think about it, for the amelioration of cold stress responses. It is a very common chemical priming agent for cold stress and freezing stress in specific. And of course, sugar polyols, as I mentioned before. We, of course, shouldn't dismiss major chaperone functions that actually includes universal defense-related molecules, such as the late embryogenesis abundance proteins, the LEA proteins. We have the core proteins, again linked with the cold responsive approaches, as well as the famous heat shock proteins, the HSPs, which is again quite big gene family. And of course, one should not forget the regulation of ion transporters, so membrane transporters, such as sodium transporters, potassium transporters, and so on. This can also include heavy metal ion transporters, but of course also aquaporins, which are the channels permeating the water transport through the membranes. So it is important to regulate water and ion movement. Through these quite complex gene activation procedures, we actually do reach a point of re-establishment of cellular homeostasis, functionality of structural proteins, as well as protection of such proteins, enzymes involved in biochemical processes, and as well as stability of the membranes, which is key to avoid further damage. And ultimately, this, when achieved, leads to the so-called stress tolerance or stress resistance, depending on the stressor, if we're talking for abiotic or biotic stressors, respectively. Okay? Is this clear? Because this is quite important to comprehend. I mean, this pathway, if you master the components, you get a very basic understanding of the main features, the main elements that are involved in the process of perception and response to the stress. Are there any questions about this, or everything is clear? Good. And of course, one should not forget that Einstein actually first mentioned that everything is relative. So what this means, because so far we're talking about concepts of tolerance and avoidance, you have to understand that what is stressful to me might not be stressful to you. Okay? And this is very basic. As an analogy, because this actually applies for all organisms, I will actually talk about myself when I first moved to Cyprus. Coming from northern Greece, I was accustomed to lower temperatures, heavy winters with snow and so on. Okay? Cyprus is a place that has much higher temperatures. As I said, it's an arid environment, and in the summertime it reaches mid-40s in temperature, so it is quite severe. So I remember when I first moved there, I had severe difficulties handling the local conditions, whereas the locals, of course, are quite accustomed to these conditions, so it really was much easier for them to actually handle this situation. Of course, this is a simplistic analogy, but it does represent the reality. Each organism does not respond the same way to different conditions. So, for example, in this case, I will actually stress your attention to specific points, such as these ones. In the case of rhododendron, which is an ornamental plant, actually, as you can see, optimal concentrations on a seasonal level for growth and development are actually ranging between 18 to 25 degrees for the summertime, whereas, of course, these temperature optima are actually lowered in the winter between 12 to 18 degrees. Okay? Now, in terms of entering a stress status, cold damage would actually incur at about 3 degrees in the summertime, whereas this actually would be about close to 0 in the wintertime. And death from freezing temperature, which is of course very dramatic, because then you have also crystallization of water, disruption through puncturing of the cell membranes, and very dramatic effects. This actually would take place at about -4 degrees in the summer, and this would of course drop to very dramatic -25 degrees in the winter. Okay? And in terms of heat stress, the heat damage is actually seen at about 35 degrees, and heat death would actually be reached at about 40 degrees. However, when we actually look at the C4 crop, such as maize, that is of course grown in very different climates and environments, the relevant optima are completely different, meaning that optimal temperature of growth would be represented between the range of 22 to 28 degrees. Okay? Heat damage would actually be achieved at 40 degrees, and death of the plant would actually occur upwards of 50 degrees, whereas the point of heat damage for the plant that would not necessarily result to its death would actually be a terminal scenario for other species. Okay? And the same stands for the lower temperature ranges, meaning that in the case of cold damage, this would actually be manifested as early as 10 degrees Celsius, which really wouldn't have a severe effect in the growth of rhododendron, for example. And likewise, the death from freezing stress would actually occur at 0 degrees, which is representative of cold stress damage for rhododendron. So everything is relative depending on the genotype, depending on the conditions, and so on. So I will keep on stressing that it is very important to address the issue of abiotic stress for sustainable agriculture, because it is sustainable agriculture. It's like a very trendy topic nowadays, but a very fundamental topic of essence. It represents a holistic approach to farming that balances environmental health, economic viability for the grower, because at the end of the day it is all about the average grower, as well as societal well-being. So abiotic stressors can jeopardize these crop yields and disrupt the food production systems, which is important for the circular economy. And we actually then by addressing these stressors, can help contribute to the broader goals of sustainable agriculture. So with this approach, we also make sure to safeguard the ecosystem at an agroecological level, because this is also important in addition to the agricultural productivity, and ultimately leads global food security and supply. So in terms of the economic implications, crop losses due to abiotic stress, of course, lead to severe ramifications. If I remember well from a recent study, in Europe alone, in the last decade, the losses due to extreme climate events was close to 25 or 30 billion US dollars. So this is like a really, really big financial factor related to the abiotic stressors I mentioned before. We also have the important element of environmental conservation, as through sustainable agriculture practices we can reduce this environmental impact of farming and thus also improve the biodiversity, which is very important for agroecological purposes. And basically, this graph actually globally represents the so-called resilience framework that actually shows the key steps we need to do, we need to follow to actually reach a point of consensus of reaching this global food security. So this, as you can see, actually spans from the understanding of the local climatic conditions that are relevant to the crops of importance for each section, because for India, the crops of importance are of course not the same as the ones that are grown, for example, in Sweden, one would say. One needs to identify a number of risks, impact, as well as vulnerabilities. So basically, it is the risk assessment approach. We have to formulate the various adaptation options, assess and prioritize these options based on importance, implement the key measures, but then, of course, it is fundamental to monitor and evaluate this effectiveness, because often we need to take corrective measures through these approaches and ultimately review the strategies so as to actually headline a key strategy at the national level. Now, yes.
So this resilience framework which you just highlighted here,
yes.
With respect to SDGs,
Mm-hmm.
I hope they play a very important role.
Definitely. This is actually directly linked with the SDGs, and it is actually a directive that, at least in the European Union, is actually promoted heavily. Right now, in the Horizon Europe call for funding, which is the main pan-European funding movement for research that actually provides many, many billions of euros of research. It is a very generous funding scheme. Actually, in the, I think it's how many, 15 or 16 DSGs, if I remember well, each call requires for you to address a certain number of DSGs. And actually, me acting as an evaluator in Horizon Europe panels, we're actually always given instructions to check that this
particular proposal is meeting.
Of course, and not actually meeting these key DSGs is a major reason for really negatively affecting the marking of these proposals. So this is a very important factor that is actually being addressed. And to be honest with you, many times the writing of the proposals, this is just confessing the reality, is always done also in a way to, in a targeted manner, try to satisfy specific DSGs. You know, other DSGs, I mean, in the European Union, and I'm sure it's the same case in India and in other nations, there is great sensitivity also with issues of gender balance, of female empowerment. There is many key important elements that are very heavily being promoted in the European Union, to the point of really strong proactive behavior, which is true, which is correct, and it should be done so. You know, so you're perfectly right, Professor Sharma. It's DSGs are tightly aligned with the proposed resilience network. This is a very spot-on comment. So, in terms now of the types of stressors, because briefly before I mentioned there is many different types, but actually we should get a better understanding of the variability of this situation. There are many ways to actually face the stressor. Later on I have a specific diagram that shows detailed categories that we'll discuss further in detail. But it is important to understand that each type of stressor represents unique problems. Of course, there are key elements that are horizontally found in the different types of stress, but actually each stress also does have specific fine points that actually differentiate it from the rest. The one thing I would like you to remember is that in terms of studying stress in the lab, I always believed, and I strongly still do so, that abiotic stress research is much more challenging than biotic stress research. Not taking anything away from biotic stress, the importance of that. I'm not saying it is not important. I am just saying that the abiotic element is much more complex. Because in the case of the biotic stressor, you usually have to face the action of a specific pathogen, of a specific insect, fungus, whatever, you know. So usually an active ingredient might actually have a point of activity that will be enough to address this biotic stressor and then result in the well-being of the plant. In the case of the abiotic stress, the response of the plant is really holistic, coordinated, and it usually affects very complex pathways. In our case, when we actually do RNA sequencing of drought-stressed samples, salt-stressed samples, and so on, usually the number of differentially expressed genes, but also proteins when we carry out proteomics, is quite impressive. I remember that recently we carried out, as a control to a priming experiment we did, of course we also have RNA sequencing of the stressed alone samples, in addition to the ones that are primed, so as to see the transcripts that are actually differentially expressed. And in the stressed alone samples of our drought-stressed Medicago plants, we had no less than almost 2,500 transcripts, and actually this keeping a quite stringent level of cutoff point for picking the specific genes. That was more than 2, I think, twofold increase. So it was a very stringent cutoff point. And actually going lower to 1.5, this number increased to something like 4,000 transcripts. So it is a very extensive network of changes being done at the molecular level. And of course, this represents bigger difficulties in trying to ameliorate this response, because it is very rarely a single point that should be addressed so as to actually reach the point of protection needed for the well-being of the plant. So, briefly speaking, of course, we have the concept of temperature stress that includes both extremes of high and low temperatures that affect metabolic processes and development. We have the water-related stress that is not only related to water scarcity, so drought, water deficit, but also is related to excess water. Flooding is actually a very important issue that actually affects heavily productivity. And it impacts, of course, water uptake heavily, photosynthetic activities, as well as nutrient transport. This is really heavily affected. And of course, with flooding in specific, you always have the major problem of reaching anoxic or even hypoxic condition, hypoxic or even anoxic conditions. So effectively, the plant suffocates. It doesn't have enough oxygen in the rhizosphere, and basically this really affects the respiration processes that actually in turn affect the production of ATP needed for metabolic reactions. So it is a very important problem. In terms of soil-related stress, of course, we have, or what we call chemical stress in general, we have the concepts of soil, of acidity, of soil salinity, soil acidity, which can also be a major issue, but of course, nutrient deficiency or excess. This can in turn disturb the delicate balance required for the optimal plant growth. Light quality and intensity is also an issue. Any changes in light quality, intensity, the photoperiod as well, and the spectral quality can severely influence photosynthesis. Nowadays there is a lot of research being done on different types of LED lighting, so as to improve productivity, especially in vertical farming systems in Europe. And generally this research is mainly linked with studies relevant to photosynthesis, studies related of course to flowering, which also affects fruit set and yield in general, and overall plant development. And one should not forget the atmospheric stress that actually faces factors like high winds that can easily lead to mechanical wounding, to mechanical damage, pollutants, this is a major issue, as well as atmospheric gases such as the ones I mentioned before that also affect plant structure. In the case of the pollutants, I am not talking just about normal chemical pollutants. One major problem that is arising lately is through the leaching of pharmaceutically active compounds in the soils or contaminants of emerging concern, mainly through the wastewater irrigation. Okay? It is quite common to actually obtain wastewater from hospital units or from all sorts of industrial scale units that through wastewater treatment are then irrigating the crops. But actually it has been seen that a big number of pharmaceutically active compounds, especially drugs of human use, actually do end up in the effluent, and this is actually taken up by the plants. There was a recent study in a global scale on rivers in all major continents, and I think that the report said that there was worrying levels of both pharmaceuticals, so mainly painkillers, contraceptives, antibiotics, of course, which is a very big problem. There was huge amounts of, actually, can anyone guess what was the biggest CEC contaminant that was found in water bodies? The most common one. It was caffeine. Caffeine was the most common pollutant that was found in the water affluents. But also drugs of a nasty nature, that are used by humans, So this is actually a problem that many people don't think about, but there is a lot of work that's been done that is showing that even in biologically relevant concentrations, this could be damaging to the plant cells, but mainly at a localized level. So, for example, in our case, we've carried out research and we published a few papers where we actually treated plants with wastewater spiked with contaminants of emerging concern, and actually we did see significant cellular damage in the root system when applied hydroponically. Thankfully, the distal tissues didn't show any damage, and there was no detection of those pharmaceuticals in the final crop. So in this case, we actually treated tomato plants, and the tomato fruit, thankfully, was not detecting any of those pharmaceuticals. But at the local root level, we actually did have damage at the cellular level. So this is quite alarming. So just to summarize the types of stressors that I mentioned briefly before, this is quite a nice table, I think, that actually categorizes the factors to both biotic and abiotic stressors. Of course, one should not forget that in the case of the biotic stressors, this might look like a small subsection, but within its subsection, there is a huge subdivision, because infection can actually cover thousands and thousands of pathogens of fungal nature, bacterial nature, viral nature. You can have mycoplasmas, you can have all sorts of microorganisms that actually infect. You have herbivory, but herbivory can also be covering both sucking and biting insects that cause mechanical damage through wounding, but also damage through the stylets, for example, of the aphids. And also we have the competition, which is dealing with invasive plant species, weeds, and other plants that lead to allelopathic events. In the case of the exogenous stressors, we already mentioned the subdivision of the temperature to heat and cold, and of course cold is submanifested to chilling and frost stress. Water, we have the extremes of water scarcity and water excess that leads to hypoxic or even anoxic conditions. Radiation is a key issue because we have the light stress, ionizing radiation that is also a factor, as well as UV related effects. The chemical stress that is usually related to the mineral salts that are either deficient in terms of low amounts, but also toxic in terms of oversupply. The pollutants in the forms, of course, of heavy metals, but also pesticides. Agricultural agrochemicals represent a major threat to the ecosystem, and of course several gaseous toxins. We have mechanical stress that can be manifested through excess wind, as I said, but also soil movement and erosion, as well as submergence. And of course we have the other stresses, which is a bit more specialized, but it also does represent a very specialized field of research in our studies, covering mainly the effects of electrical fields as well as magnetic fields. Interestingly, recently I've been seeing in the literature work on priming, where they're actually trying to utilize electric fields and ionizing radiation to actually achieve priming effects. I've even seen papers where they're now using ultrasonication for this approach, and they actually do this to functionalize plasma. So there is many different things that are currently being presenting in the literature. Exactly. So as I said, the possibilities are endless. anything that could be stressful could always be potentially promising if applied at the appropriate dose. You should always remember that. As I always tell to my students, even I mentioned this before, I think, even vitamin C, which is the major antioxidant, like when I get a cold, I get vitamin C pills, okay, ascorbic acid. Excess amounts of ascorbic acid are actually catabolized to hydrogen peroxide. And even with ourselves, actually the recommended dose of ascorbic acid according to the supplements is 2 pills, which is about 1,000 milligrams. The reality is that the organism can actually take up and utilize on a daily basis about 125 milligrams. Effectively, the remaining 875 milligrams are actually expelled through natural urination and so on. Okay? So, not all of this is needed. Excess amounts do have to be expelled in order to avoid any complications through catabolic processes. Now, if you ask me, why then do we need to get 2 pills? It's because the pharmaceuticals actually recommend this to actually increase the uptake and the sales of the product. But that's just me. It's easier to just eat a few oranges and you'll be safe. Natural way. So, the other thing you have to remember is that stressors, as I said before, represent a major complexity, mainly through to synergistic effects. Okay, when multiple stressors act together, their combined impact can be, of course, much more severe than the sum of the individual stresses. And as I said before, in points of excessive watering, flooding, you do have the stress of the anoxic conditions, but in addition to that, you will get your primary and secondary fungal infections that also will lead to further damage to the tissue. The same happens, of course, in cases of elevated temperatures. Usually this comes hand to hand with water scarcity, for example, and so on. So, the other thing, though, that you should keep in mind, that this synergism, which is a negative in terms of the stress effects, could actually be explored also in the terms of the amelioration. As I mentioned yesterday, in a similar fashion, applying priming technologies could also be explored through synergistic activity so as to achieve further improved performance by adding different priming agents. So we should always keep an open mind to look for further solutions. And for instance, as I mentioned, the combination of heat and drought does lead to much more severe attack on the cell than individual stressors. The relative damage is not identical, meaning that, for example, in my group, we've done in the past studies where we applied water scarcity, water deficit to plants in addition to mite infestation. We were using Tetranihus urtica, which is a very common mite in horticultural crops. And actually what we did see is that the combination of the stressors was more damaging to the plants, but in terms of individual stressing, the drought stress was much more damaging to the plant than it was for the mites themselves. So relative gravity of response, actually the abiotic stressor did appear to have a much more pronounced effect in comparison with the biotic stressor, but the combination of the two just actually skyrocketed the effect in terms of cellular damage indicators. So we did see clearly synergism in the effects of the two stressors. So in that sense, there is a whole array of interactive responses of the plant in response to those synergistic damaging effects. So an example is represented by when a plant experiences both nutrient deficiency, such as, for example, potassium deficiency, and water scarcity. In this case, it might prioritize water usage over nutrient uptake, showcasing a dynamic and integrated response system. And usually, the water usage is the first concern of the plant. Okay? Think in a simple fashion. When a seed germinates, the first thing that will actually arrive is the root, because the first thing it has to worry about is the water. And further on, to the nutrition through the uptake of the ions that are actually dissolved in the water. That's because it already has some basic reserves of carbohydrates in the form of starch either in the endosperm initially, but mainly also in the cotyledons. And then it can lead to the hypercotyl development, true leaf development, and then create de novo productivity of carbohydrates. The first reaction is always to go and look for water. It's the number one thing to do. So, in any case, we always have to consider potential trade-offs and prioritize these trade-offs. Plants often face trade-offs when allocating resources to cope with these different stressors. You have to understand that when a plant tries to defend itself, this is a highly energy-consuming response. The plant needs ample chemical energy, lots of ATP, to fuel all of those metabolic processes for activation of either defense responses or agronomic morphological responses by developing deeper root systems or whatever, So, in this sense, this excess energy that is required for the fueling of defenses is always at the expense of the growth. So there is always, or most likely, a trade-off. And this is why you will usually see in highly tolerant varieties of crops that the very tolerant ones are usually slightly smaller in size than the less tolerant ones. Or they might have some sort of lignification on the stem, or some elements that actually do show, comparatively speaking, more standard growth. Think about crops that are super efficient in growth, like maize. Maize is generally not considered to be a resilient crop to stressors. This is because it gives ample energy to actually have very quick growth, but at the same time this also means that its demise is that it is extremely particularly sensitive to a number of abiotic stressors. So there's always kind of a balance that has to be achieved and an ultimate trade-off. So in a scenario where a plant encounters both high temperatures and, for example, pest infestation, like I mentioned before, it might allocate resources to the heat stress response, but this might actually compromise its ability to defend against the insect. Okay? So this makes it very obvious that it is a critical aspect to prioritize the plant's adaptive strategy. See what represents the major threat and try to address that threat first. So understanding these complexities helps researchers and farmers to develop strategies that consider the interactive nature of stressors, leading to more effective and holistic approaches to what we call integrated pest management and stress management. So we always have to think big, we always have to think in a global scale. So always we have to consider the potential environmental challenges in agriculture. This is because, of course, it is confronted by many, many different challenges at the global scale. And, of course, these are linked to the diverse geographies, to the diverse climates, as well as the diverse ecosystems that define Earth. And from the ice-ridden landscapes of the Arctic to the arid regions of the deserts, each region actually has to deal with its unique stressors that represent very specific signatures of plant responses. So we also have to consider regional disparities, and in examining such regional disparities, it's very clear that what constitutes, as I said, a challenge for one region differs significantly from the other, such as with the example I mentioned before of the crops growing in different fields. For instance, drought might be a pressing issue in certain arid regions, such as Cyprus, for example, but excessive rainfall will pose challenges in other regions. And recognizing and understanding these variations is key to tailor actually these effective strategies that resonate the needs of each region in specific. So in simple words, your approach as Indian researchers should be tailor-made to your specific needs. Okay? In my case, for example, I usually reach out to the local industry and I try to figure out what problem they have. But this could not necessarily mean that it is of relevance to you, for example. And as an example, in the past we developed a priming technology that was actually being evaluated by a big multinational that was mainly linked with drought stress responses in leguminous crops. And following a lot of testing, they actually opted to go for a different technology that was prioritizing needs for improved, sunflower production in Poland, because that represented a major threat to the specific crop that was mainly affected by white mold, which is like a fungal disease of the specific crop. So they prioritized and they said, okay, we have the concept of drought stress on legumes in the Mediterranean, we have the market of the sunflower industry for the sunflower oil in northern regions and the biotic stressors, and at the end of the day there was a major prioritization, regionally speaking, that actually at the end directed the response of the company, unfortunately to the benefit of the other technology. But this is how science works. So of course at the global scale, all of these environmental challenges at the end are always translated to risks on food security. It is all about food security. With the increasing population, the drop in the cultivated land, this is becoming more and more a threat. So all of these events directly influence crop yields globally, and disruptions in one part of the world can actually trigger the so-called ripple effects throughout the global food supply chain. And this shows how sensitive and fragile our agricultural systems are. So this is why we need to act fast. So how do we do that? We have to always find for emerging solutions, and for that we need to develop innovative and sustainable solutions at the global scale, and this could include many different things such as precision agriculture or smart agriculture. I'll talk about this later on slightly. And this is very key because it utilizes IT, robotics, sensors, and smart irrigation systems to actually be able to detect stress at a very early stage, but then also do corrective measures in an automated manner. This is actually being implemented quite heavily in Europe. There is a lot of pilot trials where they're actually carrying out precision agriculture. I'm not aware, is this something that is actually being carried out on a large scale in India? Yes, good. So this, as it would be. I mean, this is something that is, of course, attracting more and more attention. Of course, we do have the necessity to develop the resilient varieties. For this, priming, of course, is only acting in an indirect manner. In this case, I'm more talking about, for example, gene editing approaches. Such as the CRISPR-Cas movement. It is quite efficient, as you're well aware. And to be honest, although Europe is quite anti-GM at present, there is a large movement to overturn the ruling that CRISPR-Cas editing is considered GM. That's called EU SAGE, and they're very close to actually achieving that. So I think we're very close to actually also allowing gene-edited crops, like CRISPR-Cas edited crops in Europe, to actually become commercially approved. So this is a clear promise. I think that this is a major factor that will play a huge role in the future. In June, I was in Japan at an international conference on Arabidopsis, and actually they were now marketing in the markets, in the supermarkets, CRISPR-Cas edited tomatoes. This could be seen, I could see them in the supermarkets. And in the States, I also saw, for example, gene-edited apples readily available in grocery stores, which was really interesting. Actually, it was a variety called Snow White, and it was nice because the concept was that they were they were suppressing the expression of polyphenol oxidase, PPO. So that way, when you would cut the apple, it wouldn't lead to the oxidation of the tissue, so it wouldn't result in quick browning, when you cut the apple. This is, of course, not threatening to the health. If you eat that, it's fine, but it's not aesthetically pleasing. And for them, it is important to actually have a nice white apple after you cut it and have it on your plate for like 2 hours. Okay, But this is something that can now be readily purchased in, large scale in the markets in the US, which is quite interesting. And of course, advanced irrigation techniques. So for that to be achieved, we need a collaborative effort between different nations, research institutes, hence all of our efforts to actually promote collaboration between countries, between groups, and so on, in order to foster the development and implementation of practices that mitigate the impact while enhancing agricultural productivity. And these solutions are promising not only for individual regions, but the collective resilience at the global scale. As agriculture grapples with the intensifying challenges of abiotic stress, it is imperative to cultivate these sustainable solutions. And it is, in envisioning the future of agriculture, it is crucial to adopt this forward-thinking perspective that embraces innovation, which is extremely important, resilience, as well as environmental stewardship. So, briefly, before I mentioned some key technologies that I think are the way forward, the innovative tools of the 21st century, and actually the concept of nanomaterials and nanocarriers for delivery of compounds is, in my book, another potential innovative tool for the future, because when we will talk about the nanomaterial technologies, it represents many ways of improving production, but also for improving gene delivery, for example. You can use nanomaterials to actually further enhance gene editing techniques. It can actually be applied on many, many different levels, in addition to the plant priming effect that we will talk about as part of this GIAN course. So first, of course, we mentioned the concept of precision agriculture that is heavily applied also in India, because it represents a technological revolution, leveraging data analysis, satellite imagery, UAV imagery as well as sensors, as I mentioned before, that are actually sensing multiple different components such as soil humidity, soil acidity, soil salinity, temperature, you name it. There are sensors for virtually everything nowadays. And this is then to supply a decision-making tool, which is an automated DSS tool, that actually will come up with the optimal solution that will then be applied in an automated fashion in the field, through the irrigation, through the fertigation, through the application of the appropriate pesticide, and so on and so on. So by precisely tailoring the inputs such as the water, the fertilizers, and the pesticides I mentioned before to the specific needs of each crop and even individual plants. Yesterday I mentioned about the use of phenotyping platforms, which is wonderful because it actually has direct actions on individual plants grown in individual pots. It is a very specialized approach. And this approach, of course, will maximize efficiency and also minimize waste while mitigating the impacts of abiotic stressors. Precision agriculture, therefore, is a cornerstone in the drive towards these sustainable and resource-efficient farming practices I mentioned. The importance of resilient crop varieties is very important, mainly through the gene editing approaches. So through gene editing, but also advanced breeding. Nowadays we have also speed breeding techniques that can actually lead to multiple cycles of a crop within the same farming system. So scientists are therefore creating, of course, crops with enhanced tolerance to specific stressors, mainly focusing, of course, to the climate change related ones, which is heat, drought, and salinity. And enhancing these resilient varieties ensures that agriculture can adapt to changing environmental conditions and thus secure reliable yields in the face of unpredictable stressors. So one should not forget that it is imperative to always remember that water is most probably one of the most important factors to always consider. So water efficiency is key for improved farming systems, especially because water scarcity is an increasing threat for agriculture. So sustainable irrigation practices such as drip irrigation and rainwater harvesting is becoming more and more used, enhancing water use efficiency. And furthermore, the integration of precision and smart agriculture tools such as sensor technologies assists further in optimizing the irrigation schedules, thus preventing both water wastage but also the detrimental effects of waterlogging conditions that lead to the anoxic conditions I mentioned before. And these water efficient techniques are pivotal for sustaining agriculture in regions that are more prone to water-related stresses. And in every case, yesterday I was introduced to the concept of biomagnification that actually affects many different levels of the agroecosystems. Agroecology is very important. One should not forget this because it embodies a holistic approach that recognizes the interconnections of farming with the natural ecosystems, which is extremely important. And by mimicking natural processes, agroecological practices can enhance biodiversity, they can promote soil health, and also create resilient agroecosystems. And these approaches include, of course, intercropping, which is becoming more and more important, and it is quite heavily promoted in the EU right now. Cover cropping with, for example, leguminous crops, and of course integrated pest management practices, thus lowering agrochemical usage. And by fostering ecological balance, agroecological methods contribute to the long-term sustainability of agriculture, thus mitigating the impact of stressors. And of course, for this lecture, I could not close without mentioning as an innovative tool the priming concept, because as I mentioned yesterday, priming offers a wonderful alternative to genetic modification, especially when this is not really acceptable, while beating the negatives of conventional breeding, which is the delayed procedures. Effective priming agents actually don't result in growth trade-offs, which is very important. So effectively, there are technologies where we do see the growth promoting effect even under control conditions, which is extremely important, because it is quite common for priming agents to sometimes lead to growth trade-offs when the plant is not necessarily being stressed. And actually, this is the holy grail of developing a new priming technology. To protect the plant itself is not so difficult. You will find thousands and thousands of papers where they show some sort of indicators where the plant seems to be protected, but it is not as simple as that. You have to make sure that it will not really have a negative impact on growth and yield and productivity, which is most often the case. Okay? So, of course, as I said, priming, I think, is a great way to go forward, especially linking the delivery of the priming agents with smart nanocarriers, as we will mention in, I think, the last lecture of this series. And as I said, more and more papers are appearing in the literature. This is an example of a book I co-edited that is exactly in the field of seed and seed leak priming, mainly focusing on chemical agents. And as an editor of various journals, actually I'm quite happy to see that we receive more and more submissions that are actually related with priming technologies. Mainly chemical priming, but lately I'm very happy to say that we are receiving a lot of submissions that are related with nanotechnology. So this is an exciting field that is booming, and hopefully it will be further supported by young scientists such as yourselves. Okay, so this is the end of this introductory small outline of the stress element in plants. So according to the schedule, do I continue straight away, or do we have a 5-minute break before I start the other one? What is the... It's okay? To start the next talk. Okay, perfect. 5 minutes. Yeah, you take it? Okay, thank you. It's the same with us. It's a theoretical minimum of 3 years, but nobody finishes at 3 years. Yeah. I wish one of my students would finish at 3 years, but no, I wish. No, it doesn't.
So I have a query.
Yes.
So I want to ask that what is the exact concentration of nitric oxide during Biotech and Biotech?
You mean within the cell?
No, sir. So
You mean to use it as a priming agent? If you remember from what I said yesterday, the first rule when you do priming is you first optimize according to the needs of your specific setup. So there is no such thing as an optimal concentration that works for everything. Usually, in common, when we use, for example, SMP, the range where we see protective effects can be anything depending on the plant species, but also the growth stage. It could range anything between 25 to 100 micromoles. So low micromolar concentrations. But there's no such thing as one size fits all. Even, for example, with NOS, aspirin, I told you, like for plant priming approaches, like when we spray the plants, we usually apply 100 micromoles. At the seed level, it works better at 50 micromoles. In cereals, spraying, the optimal concentration was 25 micromoles. So I cannot really give you an answer and say this is how much you use. But most likely, in the case of SNP at least, it is in the low micromolar concentrations, maximum 100 micromoles. With other molecules, it is more variable. Like in the case of salicylic acid, we had results that appeared to be promising with as low as 100 micromoles. Most of the literature uses a minimum of 750, and I've even seen papers where they use it in post-harvest examples, and they go as high as 3 or 4 millimoles, which is much higher than what we use. So there is also a case of the other thing I have as advice for you is don't believe everything you see published blindly. You have no idea how many times we saw papers and we tried certain things based on published works in decent journals, like not in awful journals that is like unreliable and everything. And trying to reproduce the effects that were reported, we really didn't succeed. And then we had to go back to the drawing board and do preliminary concentration gradients, and we would see that the truth was much different. So, yes, I mean, you have to do a gradient, but I would start low, probably. Yeah.
So, as you told us, like priming is okay, but smart irrigation and robotics and IT, it is like possible in other countries rather than India, because India has patches of fields, not a wide range of fields in one place, at one place.
Yeah.
I think, is it right or wrong? My opinion is actually.
Listen, every solution is ideal when and where it's possible. I mean, even in Europe, in Greece, for example, where I come from, we do practice precision agriculture, but also at a limited scale. And usually this is mainly applied in units that are actually quite, how should I call it, successful. So they have proper funding. Like the average growers and the average glasshouse producers and everything, they don't. So it's the reality also for European standards. Like precision agriculture is a great tool, but it's a very expensive tool.
Yes.
So it is mainly applied in the States, in places where actually funding is more readily available. And lately, the place that is really taking charge on that field is China. China is, they're really innovating right now in several points in agriculture.
We have one company, sir.
Sorry?
Even in India, we are having a company called as Fasal.
There you go. Exactly. I'm sure India is a big country with a huge agriculture production. So I'm sure there is many production units that apply precision agriculture. But relatively speaking, the percentage I would imagine is not that high compared to the totality of producing systems, because of the expense. In Europe, for example, the most common fields to actually apply this is in vineyards. like vineyards, wine growers, wine producers usually resort to smart precision agriculture. Yeah, because with the vineyards also you don't have huge tracts of land. You don't have like thousands and thousands of hectares to worry about. So it's more confined because it's a high premium crop, you know, high added value crop. So it is more possible in such cases. Or, for example, producing strawberries or raspberries, which is very high added value. And you can get like a lot of money per kilogram of product. So that's where it is more relevant. Now, in terms of what you said, it is not either one or the other. You also have to understand that I'm talking about solutions, so I said precision agriculture, gene editing, priming. Actually, the way forward is combinations. One of the things we're trying to do, of course, one of the things we're trying to do is like with all of our RNA sequencing analysis, we actually now have a nice database of genes that appear to be crucial in the response of the plant. So basically, we're trying to combine gene editing approaches to target specific genes in addition to providing external priming. So as to do even further improvements. So it is not exclusive. It's not like either one or the other. The way forward, I think, is actually mixing and combining the approaches. And this is also with precision agriculture. It's also the case. That is the ideal scenario, you know. So, yeah. So everybody's here. We can start again? No? Yes?
One more question.
Yeah, of course, please. As I said, I want this to be interactive, please.
So, sir, basically we are from Science and Technology students, sir. We have been working on priming on nanoparticles and even electric field and magnetic field, and also even beneficial microorganisms in field level on both yield and growing parameters, seedling parameters. So usually when we speak about priming, even we...
Which crops, what kind of crops do you use usually?
Currently buckwheat.
Okay, buckwheat, yeah. Okay.
So usually whenever we speak about priming, we majorly we focus on concentrations.
Yes.
But what we miss is the time period of what we have to do the priming, like less priming time or more even like high priming time. So even that time effect is like very bad. So even
You're talking about seed priming now.
Yes, sir. Yes,
Okay.
seed priming. So even the duration of priming is having any effects on seedling growth?
It does, but actually it's not dramatic. I mean, usually we avoid too prolonged treatments, like because if we do the coatings, it's different because you can actually play with the cross-linking of the material of the polymers to do like a sustained prolonged release. So the kinetics can be, manipulated. But if you do like direct priming with a chemical, usually we do soaking. It's the most common thing.
There was a seminar by ISTA, International Seed Testing Association, where they were focusing on the priming timing actually. So
what I was saying is that, for example, usually what we do is we go anything from 4, but not so often, to 12 hours. Like the most common approach is to do a 12-hour soak.
8 to 12 hours.
Yeah, when I say 4 to 12, depending on the species we've played in this range, the most common, as you said, is the gap between 8 to 12. And most likely we usually go for 12 in most of the cases. More prolonged cases, it might even affect the viability of the seed. So it is actually pushing it a bit too much. So we also tried in the past 24 and 48 hours, but it was really not recommended. So the timing does make a difference, but actually the opening is quite narrow. It's like 8 to 12 hours, and usually that's okay. Now, the other thing that is important for the timing is when you actually have to worry about plant application. There it is very different. For the seed priming, it's easy because you have to worry about, you don't have to worry about it basically. For the plant priming, the timing is essential. And actually, if you don't prime at the right moment, because the memory effect is lost, you lose the potential. So what we've seen, if we're going, it depends also if you're going for vegetative growth improvements, like in the case of leafy vegetables or something, or if you're going for improved yield. So in our case, for example, with our tomatoes and so on. So there it is imperative that you usually do the approach pre-flowering, because this will also affect the embryo development. So the pre-flowering approach we've seen is crucial, and the other thing we've seen, because of the loss of the less pronounced memory effect, as I called it, we actually found out that we get better results by doing back-to-back applications. So usually 2 or even 3 applications on weekly intervals up until the point of initiation of flowering. And this is usually what we've seen is what works nicely for improved yields. Now, in addition to that, we've also applied, I think you saw the example I showed you with the strawberry fruit. You can even apply it on vine during development. And the reason why, sorry? Yeah. So, yes, spraying on the fruit directly. And the reason we applied it in the small white fruit section is because we actually had the profile of the hormonal content, and actually the increase in the induction of the hormonal biosynthesis is on that stage. So there we're trying to actually manipulate also the hormonal levels, which of course are related with the further development, the ripening, and the quality attributes of the crop. And we indeed did see, as I said yesterday, improvements in all sorts of things, like vitamin C content, total soluble sugars, a whole bunch of different flavonoids. So it also works nicely like that. And also you can even apply it at the post-harvest level. But of course, post-harvest, it is mainly targeting improved shelf life, so targeting cell wall components in essence. And the number one thing you would go for there is the pathogens, the post-harvest pathogens that actually lead to the deterioration of the fruit. So timing, I think, is the number one essential point. Even the concentration, we've seen cases where we get protection even with different concentrations. Maybe better, like let's say better with 50, slightly less good with 25 micromoles, but it works with both. The timing, though, makes a difference. If you miss the timing, you miss the effect. Yeah.
So manipulating the level of any hormone or anything, like in common in India, for ripening the fruit or anything, we use to ethylene or in more concentration. So I think it is harmful for the health, I think.
Yeah, okay. You said ethaphone. Ethylene. Ethylene, yeah. Well, okay, of course it depends if you're talking about climacteric or non-climacteric fruit, but I'm smiling because when you say it is not good for the health. Yeah, I understand. At the end of the day, though, these are approved technologies that are routinely employed in agriculture. I'm smiling because I see your point, but it's a fine line. I mean, you know, some things you have to do by the book with sensitivity, but it is common practice. I mean, you know, everybody. Yeah, of course.
People used to use calcium carbide. So that was detrimental for human health. And
yeah, of course.
It was found carcinogenic.
The list of chemicals that is actually banned is changing on a daily basis, you know, and actually sometimes with disputable causes. There is cases of chemicals that it's highly disputed why they were banned, because of the biodegradation levels and so on. It depends on the eye of the beholder. Sometimes I think that in some cases, to be honest, there is overly sensitive approaches nowadays. Health is super important. I also care about my health and that of my children. But nowadays I feel like it's becoming really sensitive in certain cases, you know. So everything's relative. That's all I'm saying. You know, always keep an open mind. You know, it's yeah. And the good thing with priming is that, of course, you always can resort to the natural molecules, as I said before. Like the example I mentioned of using hormonal vitamins like vitamin C or vitamin E. Like it's very common to use ascorbic acid, you know, or melatonin, as I mentioned before, which is readily used in supplements. Who was it that said that it regulates sleep? I mean, many people use it to regulate sleep. So it is a good alternative to actually use natural metabolites that you know are, you know, for sure not really harmful for health. So you have to be smart about it. You have to be smart about it. So I think we should start because I don't think anybody else is coming. So the second lecture today is going to deal with the plant responses under stress. Professor Sharma's outline first, I think, had the signaling molecules under stress, but I feel like it is the logical course is to go more generally from the outline to the general responses, and then we go deeper on the involvement of the various different signal molecules, and then we move to the technologies, because the signal molecules, if anything, also represent a key approach to actually carry out these priming approaches. Okay? So, by the way, how many of you have a background in plant molecular biology? Just to know the...
Theoretically, but not practically.
Okay. But theoretically, you have been trained in essential plant molecular biology, yes? Yes? No? No reply. Okay. No, it's fine. It's fine. It's just to understand the level of background expertise. Okay, good. So, the important thing to remember is that plants have developed a complex biochemical and molecular apparatus. Okay? And they are really... I really get excited working with plants because they really do surprise me on a daily basis. The things that they're capable of achieving, you know. So at the molecular level, they actually possess a very sophisticated system to both perceive, process, but also respond to the various different stressors. The complexity is represented by the fact that the molecular components can actually represent hundreds, if not thousands, of genes, proteins, as well as the signal molecules. So it is not an easy task. And especially when looking, as I said, at the abiotic stress response, this is even more complex compared to the biotic stress element. So the significance of unraveling these mechanisms lies in our ability to engineer crops that can thrive in diverse and very often unpredictable environmental conditions. So with this, we're going to go a bit deeper on the molecular responses that underpin the plant stress adaptation strategies. So, as I mentioned before in the basic outline, of course, in classical molecular biology, you can have regulation of responses at many different levels. You can have a regulation of responses at an epigenetic level, which is very commonly seen when dealing with seed priming, for example, where you can have, for example, histone modifications and so on. You can have a transcriptional regulation level, which is very commonly seen, like as we saw in the example I showed you yesterday with the application of sodium hydrosulfide that actually regulated the response at the transcriptional level. You can have responses regulated at the translational level, at the protein synthesis level, and of course you can have major element is part of the PTMs, which is the post-translational modifications. And so far in literature, they have actually recorded upwards of 400 different PTMs. So as you can understand, there is a very broad range of regulatory mechanisms that can actually be employed in response to the external stressors. So the first one and the key one is effectively the alterations in gene expression profiles. And this is also linked with more general hormonal activity. Because if you study about hormonal activity, the main thing it does is to actually regulate transcriptionally the plant responses. So advanced studies reveal the involvement of key regulatory elements, and this mainly includes transcription factors, which are proteins acting upstream of the genes of interest that actually regulate their expression. This is very important to coordinate the expression of stress-responsive genes. One example that is probably the most famous one is the DREB transcription factors, the dehydration-responsive element binding transcription factors, which is pivotal in activating drought-responsive genes. And actually there is a lot of work being done where they have generated overexpressors of DREB plants for both cereals, wheat, maize, and also soybean plants. And actually there is advanced trials now in fields of the specific crops that show much improved tolerance to water deficit. Okay? So investigating the intricacies of these regulatory networks provides a deeper insight on how plants fine-tune their genetic machinery so as to confront stress challenges. So at the molecular level, the stress perception triggers a complex signaling cascade that culminates in the activation of repression, of course, of specific genes. In addition to the transcription factors that is actually involved in this regulation, one should not forget, but you can also have the involvement of co-regulators and also epigenetic modifiers. So it's a very, very big list of equipment that is actually being employed that can actually act at both cytoplasmic level, at the membrane level, at the plasmalemmatic membrane, as well as, of course, exogenously through the environment in the form of the stimulus. And this is manifested, as I mentioned before, through many, many different signal components that can either include signal ions, it can include reactive species. For the activation of these components, it can include MAP kinases or MAPKK kinases, CDPKs, and a big arsenic of the transcription factors that I mentioned before. So when the plant encounters the abiotic stressor, they recalibrate their gene expression patterns. So as an example, during drought stress, the induction of the defense-related genes is heavily correlated, it's associated with abscisic acid signaling. This is very common and this is very well known. Okay? Of course, you can also get some ABA-independent molecular responses in terms of drought conditions, but the main profile is related with ABA signaling. Okay? ABA, of course, nowadays is becoming more and more related to the regulation of ethylene, as mentioned before, because ethylene so far was mainly implicated in ripening processes, but in the last 10 years or so, more and more work is being shown to correlate the involvement of ethylene with abiotic stress responses, for sure, and also the regulation of auxins as well. So there is a very nice tight interplay between all of those hormonal pathways. Another major component that is universal in sense of appearing in virtually any sort of stressor is the regulation of the LEAs that I mentioned yesterday, which is the late embryogenesis abundant proteins. Okay? This is another component that we've seen actually shows up highlighted in virtually all of the stress conditions mentioned earlier. And of course, globally speaking, as a global response, we do usually get regulation of aquaporins, which is the channels that actually assist in the permeation of water, because the water balance is crucial for virtually most types of stress. All types of stress related to osmotic shock, but also in the case of oxidative stress, we've seen that the water balance is also part of the process. So this coordinated gene expression modulation ultimately aims, of course, to improve water use in the cell, enhance the osmotic regulation, as I mentioned earlier, and to fortify the various cellular structures. The other thing you should remember is that these are complex pathways. So it is never an easy story to tell with single components. For example, in the case of drought stress responses, the involvement of ABA actually involves the activity of several receptors, such as the PIRPIL and PP2C receptors, and this research is considered so fundamental that their identification resulted in 2 back-to-back Nature papers. So these are things that are crucial for biological sciences, and hence their importance in terms of the journals that they were published to. This in turn downstream results in multiple secondary responses, such as the activation of slack channels that ultimately lead to the phenotypic response, which is the closure of stomata. And this is what it's all about, minimizing water losses. So always, again, if you can imagine the model we spoke about before, you always have the signal perception element, the response in terms of the protein that is involved in the regulation of the reaction, And at the end, we have the physiological manifestation, which is the stomatal closure leading to the reduced transpiration levels. So there is always the similar path that is approached in every type of response, each time with different components. Okay? At the same time, though, of course, you also have other elements that are actually correlating, such as components of protein and membrane stabilization. So very complex networks. So, as I said before, the genes that are actually regulated in response to these stresses encode proteins that act as effectors that orchestrate a cascade of events that ultimately aims to fortify the plant against environmental challenges. And that's why it is very crucial to gain deeper mechanistic insight. Yesterday was mentioned by one of the attendees that mechanistics is key to obtaining deep insight. Because when you study the stress, you can always just correlate with key elements, such as phenolic levels, or sugars, or I don't know what. This is all correlative. So it is effectively descriptive. It does not give functional essence to the reaction. For the functional essence of the reaction, you have to have functional evidence for the activity of specific transcripts or specific enzymes that are actually related with this response. Otherwise, it remains descriptive. This is an example of a review that was published in Trends in Plant Science, just to show the complexity that is correlated with stress responses linked with the auxin metabolic pathway, which is extremely important in this reaction. And auxin is, of course, also important when we're looking at growth promoting technologies, when we use those priming technologies as growth promoting approaches under control conditions. Because, as you know, auxin promotes the cellular elongation through the activity of expansins, XETs, and so on, through the proton motif force created by proton pumps. So this is not to intimidate you, just to show you the complexity. That in this case, you can see that we have a huge number of transcripts that are actually activated both in a positive as well as negative manner. This is, of course, done in the activity of the polar transport active fashion of the auxins. And in addition to that, we have major regulation of transporters of auxins as well, which is effectively the PIN transporters. We have large family of PIN transporters that are all involved in this remobilization of the hormones. Another example of such complex mechanisms that is related with ionic hyperosmotic shock, so effectively salt stress responses, is the so-called salt overlay sensitive pathway that I mentioned yesterday, the SOS pathway. This is another key pathway that is directly linked with hyperosmotic shock, and it is critical for the response to plant stress. And it is generally organized by 5 different components. 3 of them are essential for the function, SOS1, SOS2, and SOS3, and SOS4 and 5 appear when needed, their activity. So effectively, SOS1 encodes a plasma membrane sodium antiporter. So effectively, this is a plasmalemmatic transporter that directs sodium away so as to reduce toxicity, and actually introducing within the cell protons that have been exported with the use of proton pumps. Okay? Of course, in addition to this plasmalemmatic sodium antiporter, you can also have sodium transporters on the vacuole membrane, which is called the tonoplast, that actually introduces the sodium ions in the vacuole. And this is done because the vacuole is considered to be a detoxifying organelle. So there is different ways of actually harnessing this. It's either excluding from the cytoplasm by external pumping, or through inclusion in the vacuole with the tonoplastic transporters. Okay? So it actively transports sodium out of the cells to maintain the cellular homeostasis, or it can actually compartmentalize it within the vacuole again to reduce the toxicity effects. And this ultimately prevents the toxic accumulation of sodium. And another example is RD29A, which is a gene that is regulated by the previously mentioned transcription factors, and specifically DREB1A. So again, here we have the regulation of a directly regulatory transcript for the response to saline environments. DREB, of course, is also linked with arid environments, with water scarcity environments, and this is actually regulated by the specific TF. And RD29A in itself is actually an LEA itself. It is a late embryogenesis abundant protein that contributes effectively to cellular osmoprotection and as well as drought tolerance. And this is why it is implicated in both drought and salt stress responses. So, of course, we have the activation of multiple molecular signaling pathways upon stress exposure. So, at the forefront of stress responses, we have intricate molecular signal pathways that transduce the external stress cues into a cascade of internal responses. And these pathways act as a very sophisticated network of communication, where they actually crosstalk with each other, and this enables the plants to perceive, process, and respond to stressors in their environment. So when the plant perceives the stress, first it does so at a cellular level. So we have receptors in the cellular membrane, in the plasmalemma, that or within the cytoplasm, that can actually recognize the specific stress signals. And this can also be through recognition of changes of concentration. The sensitivity is actually extremely high. This can actually go through also sensing of reactive oxygen species alterations, because as I mentioned yesterday, although in high levels we do result in oxidative stress, but in low levels this acts as a major signal transducer. Of course, we also have the alterations in hormonal balance, which is again very low molecular concentrations, but any fine differences can actually be easily perceived by the receptor proteins to activate the defense responses. So this initial recognition triggers a series of intracellular signaling events, and as an example, in response to drought stress, ABA again serves as a central signal molecule that basically results in the activation of several defense components. And here we have one example of what is happening at a molecular level in a salt-stressed cell, and you can see that the network is huge because it involves, as I mentioned before, several plasmalemmatic transporters that could either be ion transporters such as SOS1 that, as I said before, pumps sodium outside the cell by bringing protons inside. The AHAs are basically proton pumps, and proton pumps are very important because they are primary transporters. The secondary transporters, which are the ion transporters, rely on the generation of the proton motif force, an electrochemical gradient across the membranes. This gradient is generated by the proton pumps that actually pump protons outside the cell. And this also creates an acidic environment at the apoplast, which is also important for the activity of several enzymes, like the XCTs and the expansins that are related to the cell expansion linked with auxin activity. So all of these things are interconnected in some way or another. And as you can see, we also have activity of several other components, such as MAP kinases and so on, that at the end of the day all function towards a similar goal, which is to create a homeostatic status in the cell to achieve protection against salt stress. And this is the case of the potential function of the sodium transporter that I mentioned, is tonoplastic, and actually if some sodium ions escape the function of these transporters and they remain in the cell, this then comes to collect what is escaping and then compartmentalize within the vacuole. So quite a smart way of actually protecting itself against the stress, but through the regulation of several molecular components. It's not easy to achieve so. So these pathways often involve protein kinases. It often involves phosphatases, like the PPLC that I mentioned before, and the secondary messengers, such as the calcium ions that I was mentioned to before by your colleague, which relay and amplify the stress signal within the cell. So these pathways, which are collectively known as signal transduction systems, culminate in the activation or repression of downstream target genes. And understanding these very complex signal networks is very important to decipher how plants integrate and process stress signals to mount an effective response. So in this response, of course, there are many components like the reactive species and the specific ion signals like calcium, but of course the involvement of hormones is fundamental for these activities. It's crucial. So hormones serve as key messengers in conveying and coordinating these stress signals. And these molecular regulators play a pivotal role in integrating the diverse stress cues and tailoring the response of the plants under the stress conditions. So, hormones. What kind of hormones come to mind in plants? Let me know some of the major hormones that you can think of. Auxin, gibberellin, cytokinins, correct. Yes, ethylene, as mentioned before. In addition to the 5 major ones, like the initially studied ones, like the initial group of major hormones was, as you mentioned, auxins, cytokinins, which are the essential hormones. When we say essential hormones, we mean that we cannot have viable mutants of these hormones. So we cannot have mutants that don't produce auxin and cytokinin, and to have a plant that will complete the life cycle. It can survive up to a certain stage, but it cannot complete the cycle by producing seed. This is what an essential hormone is. The same way, this is what an essential element is. It cannot complete the life cycle without it. Okay? So, auxin and cytokinin are the 2 essential hormones that you cannot find viable mutants of. And the other 3 initially important hormones were, as you mentioned, ethylene, gibberellins, and the abscisic acid, of course. Now, in addition to those, a lot of more extra molecules have appeared throughout the years that know or exert hormonal activity, and you mentioned a few of them. I heard somebody mentioning salicylic acid. Brasinosteroids are also very important. Jasmonic acid, which is heavily linked with the insect responses and the wood effects. Strigolactones, which are actually very linked with root activity, and they're actually very heavily researched lately, to be honest. But in addition to those components, there is more. The list is actually ongoing. Yesterday I mentioned polyamines. Polyamines are known to act as hormonal signals. Melatonin. Melatonin is actually nowadays considered to be a new, within brackets, phytohormone, especially because A, it acts in a similar fashion to hormones, and B, structurally speaking, as we said, it shares a very common backbone with auxin. Okay? And of course, there is other cases of signal molecules that show hormonal activity. Lately, I've been reading about the importance of carychins, which is smoke-derived molecules that actually also regulate growth. So the list is ongoing. It doesn't stop there. You know, what we considered the major hormones, this is actually becoming enriched as time goes by. So these are considered the master regulators in the regulation of these stress responses. And each hormone is generally associated with specific stressors, but overall, you should bear in mind that usually most stressors will have some sort of effect on the regulation of most of these hormones. It's just that some of them play a more important role. Like, for example, in your mind, the main hormone that comes to mind if I say the words drought stress, everybody will talk about ABA, for example, you know. Or Even for the ripening process, there is some physiologists that consider ripening as some sort of stress for the cells, in a way. Of course, personally, I'm not sure I agree with this assessment, because it is just a natural development process, the same way that senescence is a development process. You know, I wouldn't call senescence a stress per se. It is stress because the cells are actually affected, but it is a natural stage of development. But of course, this is disputable. People can have their own opinions, you know. So yeah, each hormone is relatively more correlated with specific conditions. And ABA, as I mentioned before, the main reason everybody thinks of ABA when it comes to drought stress is the main end product of ABA activity is, of course, the stomatal closure, the regulation of the stomatal movement, which is directly linked with the water status of the cell. Ethylene is very commonly associated with the concepts of senescence, as I mentioned before, but mainly also pathogen attack. Lately also, there is a great correlation with drought and salt stress in particular. And as mentioned already, salicylic is correlated greatly with pathogens, bacterial and fungal, and jasmonic acid is greatly related with herbivores and generally wounding responses. So overall, we do have the implication of specific pathways. Again, I will repeat that these are not exclusive of others. For example, if you remember yesterday, I mentioned that the application of NOS aspirin in plants under drought stress, in the RNA sequencing analysis, let's see who was paying attention. What was the main family of genes that was upregulated following application of NOS aspirin in plants under drought stress? Does anybody remember? Sorry? What was it?
BHLH.
BHLH was involved in the strobilurins. I know it was a heavy lecture yesterday, so it's okay, but it was lipoxygenases, the LOXes, the lipoxygenases, and actually specifically 9 lipoxygenases, because there is a big family consisting of 13 lipoxygenases and 9 lipoxygenases, and one of them uses linolenic acid as substrate. The 9 lipoxygenases use linoleic acid as substrate, and actually in this case it was the 9 lipoxygenases, which is effectively LOX1 and LOX5 in Arabidopsis specifically. Now, lipoxygenases are directly implicated in the past with jasmonic acid, which is linked with the so-called response to herbivores and wounding. And yet, here we are identifying that the major family that was actually upregulated following application of NOS aspirin under drought stress conditions was LOX. So now we are suggesting a new function of the jasmonic pathway in relation to drought stress responses. So again, this comes with my advice to always keep an eye open for new developments. Don't think in your mind JA is linked with insects. I don't work with that, so this is out of the question. You'll be surprised at what kind of new discoveries you might make that are not really recorded in the literature. And this is what makes innovation exciting. Whoops, sorry. No, I did something wrong here. So this is just a general representation to summarize kind of the points I mentioned before, which is the plant exposed to multiple stressors, both of biotic and abiotic nature. That leads to the concept of singular transduction through various components like calcium sensors and so on, and also through the transduction of signal through the regulation of hormonal pathways such as the main hormones that we mentioned before. In addition to the transcriptional regulation, the other major element that appears to have a role in the stress responses is the PTMs, is the post-translational modifications. And for that we mean when a mature protein actually becomes modified post proteinosynthesis. Okay, so this could cover, as I said, various PTMs that the list I think goes up to 400 different ones that can range from glycosylation, to carbonylation, to tyrosinitration, to S-nitrosylation, to phosphorylation, and so on and so on. And with the post-translational modifications, this is a great way to trigger the activation or deactivation of a protein. For example, with the phosphorylation of proteins, we usually deactivate their action. And to regain activity, we have to phosphorylate again. And so on. You know, oxidized proteins, which are damaged proteins, become carbonylated. And this reduces their activity. So there's many ways of actually modifying proteins at the post-translational level, but this has a direct effect on their function. So these PTMs are critical players in shaping the functional landscape of proteins during stress adaptation in plants. And specifically with priming, we've seen that the PTM element is key for the activity of most of these molecules that I talked about yesterday, and I will talk about further in the following days. So these modifications, another example that I forgot to mention is ubiquitination, act as dynamic regulators that fine-tune the activities and stabilities of proteins involved in stress responses. So PTMs confer an added layer of complexity to the regulation of the stress-responsive proteins, and in response to stress, specific amino acid residues on proteins are the ones that undergo chemical alterations. So for the example of phosphorylation, the addition of a phosphate group can activate or deactivate proteins, mostly the latter, involved in stress signaling pathways. And ubiquitination mark proteins for degradation, regulating their abundance in the cell. And glycosylation, for example, which is the addition of sugar moieties, can modulate protein stability as well as function. So a big array of modifications. And stress proteins, including molecular chaperones like the HSPs that I mentioned earlier, can actually act as pivotal players in this stress adaptation. So they assist, of course, also in protein folding, they prevent aggregation, and they facilitate the degradation of the damaged proteins. So the proteolytic element is also very important, because that way you can also get de novo synthesis of the free amino acids that can then be redistributed to create new proteins, new functional proteins. So proteolysis is also very important. Okay? You have many different compound elements now that are actually very trendy in literature, like the activity of metacaspases and many different things. Autolytic, autophagic pathways and so on. So PTMs play a crucial role in regulating the activities of stress proteins, and this ensures that they will function under stress conditions. So in terms of the role of stress proteins in cellular protection, such as the role of chaperones, like the HSPs that I mentioned before, they act as guardians, one would say, in the cellular arena, playing a pivotal role in protecting the structural and functional integrity of proteins during times of stress. And these molecular guardians are key components of the cellular machinery, ensuring proper protein folding, and the components that I mentioned before. HSPs are a huge family. I think in Arabidopsis it is represented by more than 40, 50 odd isoforms. So we're talking about a very complex network. So stress proteins come to the forefront when plants face environmental challenges. Chaperones basically mainly work in the unfolding of the proteins, of denatured proteins, and preventing the formation of non-functional protein aggregates. And beyond folding assistance, they also contribute to cellular protection by guiding the degradation of the irreversibly damaged proteins due to oxidation, nitrosylation, and so on. So this quality control mechanism ensures basically that malfunctioning proteins do not accumulate and compromise cellular function. So HSPs, which is a very well-known family of chaperones, are often correlated and induced during stress and as central players in this cellular protection mechanism. So in addition to the LEAs that I mentioned as being universal stress regulatory components, another universal component would be considered the HSPs, although their name would suggest that they're linked directly only with heat stress responses, we've actually seen that HSPs are actually regulated, some form of HSP anyway, through virtually any sort of stressor. That's representing a universal regulatory component. So you can see here a diagram that shows the overall functionality and importance of the HSPs in particular, the heat shock proteins. So they assist in the folding of newly synthesized proteins. They guide translocation of proteins across organellar membranes and intercellular compartments. They prevent disaggregation of oligomeric proteins as well as unfolding. They facilitate proteolytic degradation of the unstable proteins that are usually resulting from the damage due to reactive oxygen and nitrogen species excess. And also they mature signal molecules and transcriptional activation of transcription factors. So as you can see, a broad array of functionality that is important for the response of the plants to the stress. So how do plants reconfigure these metabolic pathways to actually handle plant stress? So the response of plants to stress involves a deep reorganization of their metabolic landscape, a strategic shift in the utilization of resources to navigate the challenges posed by the stressful environment. So effectively, one of the first things that is always affected is photosynthesis, because the main thing that you have to remember is, as we said before, the plant needs energy to fuel defense, and this energy is most usually originating from carbohydrate resources. Okay? We've seen that in most cases of successful priming, the main components of the proteome that are actually improved are usually linked with photosynthetic capacity. So this underlines the importance of this element. So this metabolic adjustment is a dynamic and intricate process that involves the fine-tuning of biochemical pathways to ensure the plant's survival and adaptation under these stress conditions. And as you can see, it is a very diverse subject. Plant metabolism is a huge field because it covers both primary and secondary metabolism. By talking about primary metabolism, of course, we're talking about sugar production, carbohydrate production, as well as amino acid and organic acid production, which is involved for the proteinosynthesis element. But in the secondary metabolism, there has been identified in excess of 40 to 50,000 different metabolites from plant origin. Of course, I don't mean each plant produces 50,000 metabolites. This is in total in the plant kingdom. Okay? But it is a huge source of secondary metabolic material. And this is, of course, also very useful in the pharmaceutical industry, in the cosmetic industry, in the food industry, and so on. Okay? Due to their function. And in most cases, the secondary metabolites are mainly linked with protection against stress. Plants produce alkaloids to protect themselves. Think about common examples that everybody knows. Urtica pilulifera is the Latin name for, I don't know what's the common name, nettle, like the thing that if it touches you, it makes you scratch, itch, you know. Basically, this is caused by the excretion of acetylcholine, which is a secondary metabolite that is secreted from glandular trichomes. So what happens is that when the animal will touch the plant, it will break off the head of the glandular trichome, acetylcholine will actually be excreted, and this is what causes the reaction with the skin to make you itch. And the result is the grazing animal will get annoyed and it will never go back to the same plant. So this is a mechanism of protection. You know, the most common thing, I mean, I assume you eat chili peppers here, like hot peppers, you know. The essence of this spiciness is an alkaloid that is called capsaicin. Capsicin, how do you spell? We call it capsaicin in capsicin. Okay. So, and even they use this capsicin in Greece anyway. We have products where we have like some sort of plaster that you put on your skin that is infused with capsicin when you actually get a sore back or whatever, to actually relax the muscles. Capsicin, do you know how it's generated? The plants produce capsicin because it is an antifungal agent. it is antimicrobial. And basically, chili peppers in nature almost never get fungal infections. This is because of capsicum. So, I don't know if you're religious, but God has created nature in a truly wonderful way. You know, everything is there for a reason. So, the defense-related components covers many, many different things. You have alkaloids, you have phenolics, you have flavonoids, you have all sorts of wonderful secondary metabolites. We have terpenoids, and terpenoids are also very important because they're the only class of gaseous secondary metabolites. Okay, and you have a vast range of terpenoids. You have monoterpenoid compounds, diterpenoid compounds, triterpenoids, sesquiterpenoids, and all of these are also precursory molecules for many other things. All of the hormones actually arise from forms of terpenoids. So this is a very complex network that is somehow interconnected in this response. Okay? Polyphenols are known to be antioxidants. Okay? It's a very common feature. The terpenoids, which are the precursors of hormones and vitamins, they also act as antioxidants. And of course, they also influence organoleptic parameters, quality parameters. So with polyphenols, this affects the firmness of the fruit, the bitterness. Alkaloids are linked with that as well, of course, the color, and so on. And terpenoids are also linked with the smell, with the color, with other attributes. So all of these are heavily interconnected with the stress responses. So metabolic adjustments during stress encompass a broad spectrum of changes in multiple biochemical pathways. And one very key adaptation involves the synthesis of the osmoprotective molecules that we mentioned before, the osmolytes. And these include the sugars, the polyols, but mainly glycine betaine and proline. Proline is an essential amino acid, but it is the first thing that will be upregulated when the plant is under hyperosmotic shock. And this is why we use proline content as a key cellular indicator if we carry out salt stress and also drought stress experiments. If you do a salt stress experiment, if you want the key indicator to check that your setup is working, quantify the proline. If it doesn't go up, something is wrong. Simple as that. Okay? So these compounds all play a crucial role in osmotic regulation because they maintain cellular turgor and they prevent dehydration during water stress. So in addition, stress often triggers the activation of the antioxidant pathway that leads to the synthesis of molecules like glutathione and ascorbate, as we mentioned yesterday, which are non-enzymatic antioxidants. In addition to that, you have other molecules with similar activity, like vitamin E, like tocopherol. And these act as molecular protectors against oxidative damage induced by the stressors. And energy metabolism also undergoes major reconfiguration with a focus on maintaining an adequate supply of ATP. It is essential that we have enough chemical energy to fuel respiration. Okay? This is mainly trying to safeguard production of adenosine trisphosphate, but also NADPH. And because NADPH is a redox analog that is also needed to fuel reactions. NADPH, for example, is essential in the light reactions of photosynthesis. And the list goes on. So this is achieved through adjustments in pathways like glycolytic pathway and the TCA cycle, the tricarboxylic acid cycle, which is of course the second step in the cellular respiration, as you know. And furthermore, secondary metabolite pathways may be of course excuse me, activated, thus contributing to the synthesis of specialized compounds that assist in the stress defense. So here again, this is taken from a review article that actually summarizes different concepts that were mentioned before. The plant is exposed to the stressors, so this will have a major effect on both primary as well as secondary metabolism. So in response to that, we have the involvement of sensors and the relevant responses. That includes the concepts of photosynthetic regulation as well as morphological responses. For example, before we said that a major morphological response in water scarcity environments is for the plant to try to get deeper root systems. So the root system architecture will actually be modified. Plants that are exposed to high UV conditions, they will actually develop thicker cuticles, the epicuticular wax, so as to protect itself, and also this secondarily also helps with reduced transpiration, lowering loss of water. So there is a lot of phenotypic morphological changes that might actually be happening. This is not only in cases of such stresses, this is also in cases of suboptimal conditions, such as for light. You have the growth of shade-tolerant plants. Shade-tolerant plants can actually grow very nicely, for example, under the canopy of tall trees. How do they do that? In addition to biochemical alterations, mainly on the analogy of photosystem 1 and photosystem 2 centers in the cell, they also have, for example, much thinner mesophyll. They are much thinner. And this is logical because that way it's easier for every single photon that will escape to go there to actually excite the pigments in the cell. Whereas if you have a very thick mesophyll, it will be much more difficult. You know? So very clever morphological adaptations. Of course, we have the concept of the transcription regulation, the stress-induced gene regulation, the antioxidant defense that is both enzymatic as well as non-enzymatic, the osmolytic accumulation, which is essential for high superosmotic shock, the ROS scavenging, which is linked with the antioxidant defense, and the drops in the lipid peroxidation. Of course, one should not forget the importance of phytohormonal signaling. That is usually for the tolerance linking increases in auxin and cytokinin as well as gibberellins, whereas in the case of ethylene, it's a different story. We usually try to have alterations on a negative level for the ethylene. Abscisic acid actually goes up in terms of the stress response, so as to actually lead to the closing of stomata. And of course, you can also have other effects that are actually affecting the rhizospheric microorganisms. So this includes improvements in nitrogen fixation through rhizobia, which are nitrogen fixing bacteria, like Azotobacter, for example, the solubilization of phosphorus, and the uptake of phosphorus is also mainly assisted by what? You know what really helps with improved phosphorus uptake? Yes, exactly, mycorrhiza. This is the best way to improve phosphorus nutrition. When the plant is young, it can be actually colonized by AMF, and what happens is that the hyphae of the fungus actually replace the development of the root hairs. The root hairs. Because the main uptake of water is done through the root hairs. 90% of the water is done through the root hairs, you know, in the elongation zone. So basically, the hyphae of the plant, of the fungus, replace the activity of the root hairs. And in the same way, if a plant is mature and has developed the root hairs, it is then not colonized. It cannot be colonized by AMF, unless if you overload it with a really big inoculum. But naturally speaking, then it's not really possible, you know. And this mainly affects the uptake of the phosphorus element. Of course, you have the involvement of VOCs, of the volatile organic compounds, and the gaseous elements in the response are becoming more and more important in studies. You have phytochrome modulation, which is also linked with light activation, and you also have the involvement of siderophores for iron and exopolysaccharides. And of course, at the end, this is always translated to improved yields, improved biomass, and ultimately stress tolerance. So there is a number of key metabolites that play a major role in this response. And these metabolites can act, as I said before, as signal molecules, osmoprotectants, as well as antioxidants. And as you can see here, as a basic plan, the primary metabolism is mainly linking, as we said before, the photosynthetic and the respiratory pathways that lead to the production of the carbohydrates and the chemical energy in the form of ATP. Basically, you also have the TCA cycle in the respiration that is involved in the production, in the usage, of the organic acids. And you also have metabolic intermediate biosynthesis. And in the secondary metabolism, you have the phenolics that are biosynthesized, the alkaloids, as well as the flavonoids. And from these, you also have the activity, the biosynthesis of the terpenoids. And in addition to all of these components, this result in effects in a whole array of processes, such as pathogen invasion, light responses, mineral nutrition, abiotic stress responses, antioxidant system activation, induction in photosynthetic apparatus, defense responses, as well as stress responses and signaling. So before I mentioned the major osmoprotectants, the compatible solutes, as we mentioned. So specifically, we talked about proline, which is an essential multifunctional amino acid. Its accumulation during stress helps maintain cellular turgor, as well as stabilize proteins and cellular structures. And it also exhibits antioxidant properties that contribute to the plant's defense. So as I said, especially in terms of hyperosmotic shock, you should always look at this as one of your first indicators for successful stress imposition. So you have soluble sugars such as glucose and sucrose that also act as osmoprotectants because they contribute to osmotic adjustment, leading to osmotic homeostasis. But they also act as energy reservoirs because they provide the necessary fuel for cellular processes and osmotic regulation. You have polyols such as mannitol. Mannitol is commonly used in vitro experiments, or sorbitol, for example, because this can also lead to hyperosmotic shock, like in the case of polyethylene glycol. It's a very common tool to use for in vitro hyperosmotic testing. So polyols like mannitol act as compatible solids as well, aiding in the osmotic regulation and preventing cellular dehydration. And they also possess antioxidant properties as well, in a similar fashion as proline. And one should not forget glycine betaine, GB, which acts also as a powerful osmoprotectant, regulating responses to stress and contributing to osmotic balance. It plays a key role in stabilization of proteins and membranes, and this is mainly implicated in low temperature stress. So in terms of successful priming approaches, usually the best priming approaches with GB are done towards cold and freezing stress tolerance. Okay? And actually, now we're considering functionalization of nanomaterials with glycine betaine to actually have better delivery of GB for improved cold protection in a project we're running in Cyprus on cold stress priming of potatoes. So this is, again, a diagrammatic presentation. By now you understand I like diagrams because it makes it easier to comprehend the basic concepts. So you have basically the initial exposure of the plant to the individual or the combined stressors as commonly seen in natural conditions. This leads to the activation of the osmoprotection defense system as previously discussed through the protection of cellular structures, ROS scavenging, the protection of the photosynthetic apparatus, and the protection of the enzyme stability. And this ultimately leads to protection against stress and thus stress tolerance. So for the antioxidants, as mentioned before, we have the enzymatic and non-enzymatic ones. So for the non-enzymatic ones, as Professor Sharma said yesterday, glutathione is a key antioxidant because it plays a crucial role in detoxifying ROS generated during stress, and it participates in redox reactions. Hence, the redox status of glutathione is important for stress responses. This is effectively the ratio of GSG to GSSH, for those of you who know the terminology. It helps maintain cellular redox balance, and it also protects membranes and proteins from oxidative damage. The precursor molecule is lambda cysteine, and this is interesting because this also acts as a precursor molecule for hydrogen sulfide. So there is again common elements and protective mechanisms, like in many other cases. And of course, a very important antioxidant is vitamin C, ascorbate, ascorbic acid. That is a very potent antioxidant that directly scavenges reactive species. And it also regenerates other antioxidants like glutathione. It acts as a substrate for enzymes such as APX, ascorbate peroxidase. And ascorbate peroxidase is a very important enzymatic antioxidant because it's also highly compartmentalized. You have forms of cytosolic ascorbate peroxidase that are highly detoxifying in the cytosol, but you can also get stromal APX, thylakoid APX, many different isoforms. So highly compartmentalized. Whereas catalase is usually located in the peroxisomes. It's more localized. And it amplifies the cellular defense against oxidative stress. Again, the redox status of ascorbic acid is very important. And this is effectively the ratio of ascorbate to dihydroascorbate, which is the oxidized form. Okay? I will repeat though in saying that the catabolism of ascorbic acid also acts in a pro-oxidant fashion because it leads to the production of reactive oxygen species. You should remember that. And here in the model you see the various components that cover both non-enzymatic antioxidants as well as enzymatic ones. So then we talked about energy-related metabolites. So one case was, of course, ATP, which is the universal energy molecule of cells, because it powers a multitude of metabolic processes. And during stress adjustments in energy metabolism pathways, such as in glycolysis and the TCA cycle, ensure a sufficient supply of ATP to drive essential cellular functions. As you can see, these are the first two steps of the respiration pathway. Okay? And it's interesting to remember that glycolysis happens in the cytoplasm, whereas the TCA cycle then moves on within the mitochondrion. Okay? And of course, at the end, you have oxidative phosphorylation that is the major producer of the ATP molecules. Okay? So it is also happening in different compartments of the cell, not just the mitochondria. And of course, you also have the involvement of nicotinamide adenine dinucleotide phosphate, or NADPH, and it is crucial for many redox reactions, including the light reactions in photosynthesis and biosynthetic processes. And its availability is often linked to the pentose phosphate pathway, which is essential to maintain, again, cellular redox homeostasis and to combat oxidative stress. Okay? So here, for example, you see, what do you see here? Who recognizes this chain here? Let's see your basic plant physiology. Who recognizes this? What does this represent? Excuse me? It's light oxidative phosphorylation. This is the light reactions of photosynthesis, effectively. The oxidative phosphorylation is similar in principle at the last stage of respiration and in the light reactions of photosynthesis. This is the light reactions of the photosynthesis that happens in the thylakoid membranes of, you all know this. Good. Where you have the direct linear movement of the protons connecting photosystem 2 to photosystem 1 with the in-between function of cytochrome B. And of course, you have the mobile components of plastoquinone and plastocyanin. And at the end of the day, you also have the fueling of the ATP synthase that actually produces the ATP, in addition to the NADPH that is produced in the ferredoxins. So, very important pathways that lead to the production of these energy analogs. Okay? But you're all aware of this. And in terms of the secondary metabolites, which is again a vast library of materials, it includes materials like flavonoids, terpenoids, and alkaloids. Flavonoids are secondary metabolites that contribute to the stress defense by acting as direct antioxidants. They act as UV protectants, very, very commonly, and also signal molecules in plant microbe interactions. and this also includes the category of flavanols, anthocyanins, the whole flavonoid pathway that is heavily implicated in stress responses. When plants are exposed to stress, flavonoids really increase. And for example, a main example of flavonoid research for stress is with grapevines. The most research done for flavonoids is usually related to grapevines under stress, because there you have a whole series of flavonoids that are actually and anthocyanins that are upregulated. And usually, what is interesting to know is that for grapevines, for example, drought stress is beneficial for the quality of the fruit, because it leads to the increase in the content of these flavonoids, and thus by obtaining the crop and eating the grape, you actually get better protected through their antioxidant activity. So in a way, for certain crops, stressing them helps their antioxidant profile. And this is a key example for grapevines. We've actually done research on a similar subject where we actually drought-stressed Shiraz grapes in Greece, and we did the profile of all of the flavonoids as well as the molecular pathway. We characterized 13 genes that are involved in the process, and we're actually having a paper under review in a journal exactly showing this component. They also act, as I said, in signal molecules in plant microbe interactions. Terpenoids are very diverse in structure and function. We already talked about this. They participate again in stress responses by serving as antioxidants, as antimicrobials, as mediators of plant interactions with the environment. And especially when I'm talking about metabolites that are exuded through glandular trichomes, they're always in the form of terpenoids, always. Like labdanoids, for example. And alkaloids have roles in stress defense. We mentioned the example of capsicum, acting as deterrents against herbivores, against pathogens. But also some alkaloids possess antioxidant properties. They're usually linked also with bitterness. Sorry? Yeah. They're also linked with bitterness sometimes, and that's why they say that the more bitter the vegetable, the rocket or the whatever, the better it is for your health. Yeah, of course. And here you see again a model that I got from a relevant review that actually examined the effect of the stresses to the metabolic pathways that, as you can see, leads to the regulation of many transcripts. These transcripts that you have here, they're all linked with the flavonoid pathway. And also it regulates enzyme activities like PAL, CHS, shikimate dehydrogenase, polyphenol oxidase that I mentioned before in the snow white apples and so on. And this leads to the enhanced biosynthesis and accumulation of phenolics, enhanced raw scavenging, and ultimately improved stress tolerance. So, in terms of epigenetics, because we shouldn't forget about that, and epigenetics is especially crucial when considering seed treatment approaches, because there is ample evidence that seed treatments for priming, for example, really does appear to lead to significant epigenetic modifications. And this is what leads to this intergenerational memory effect that can often be translated to transgenerational effect. There is also papers that are published recently that show potential transgenerational inheritance of improved traits, which is wonderful. There is a very well-known paper published by Anna Amtman's group in Glasgow that showed that mildly stressing Arabidopsis plants against drought resulted in the next generation of plants obtaining the seed being highly tolerant to salinity. This is one example. This was attributed to key epigenetic modifications that were actually identified through ChIP-seq analysis. So this is something that we have seen in the literature. So as plants confront these stressors, they employ a sophisticated system of molecular modifications that are known as epigenetic ones. And epigenetic changes basically encompass mainly DNA methylation, histone modifications, with histones being proteins that are structurally and functionally protecting the chromosomal DNA, and small RNA molecules that serve as a layer of gene regulation that goes beyond the traditional genetic code. So this extra layer of epigenetic control provides plants with a dynamic inheritable means to adapt their gene expression patterns to cope with environmental challenges. And this again shows similar epigenetic modification diagrams that shows how epigenetic variation can lead to change gene expression in the long term, inheritable expression levels that go to transgenerational effects, but also short-term transcriptional alterations that lead to within the generation's survival. So in the cases of the different potential epigenetic modifications, first of all, the most common one that is usually examined is DNA methylation, which is a chemical modification where the methyl group is added to the cytokine base, often occurring in a CG content. So that's why we usually look at the CG percentage of our islands, the so-called CG islands within the genome. This modification can influence gene expression by modulating the accessibility of the DNA code. So any changes in DNA methylation patterns can activate or repress specific genes, the same way that we talked about in the PTM situation, for example. And under stress conditions, alterations in DNA methylation act as a regulatory mechanism that shapes the genetic responses of the plant to the specific stress challenges. Then we also have the concept of histone modifications, which involve alterations to the proteins that surround the DNA, including acetylation. This can include methylation, but also this can include phosphorylation. Okay? Most commonly in papers examining histone modifications, people will mainly examine methylation patterns or acetylation patterns, not so much phosphorylation patterns, linked with priming, I mean, mainly. And in papers I have read, I have seen works linking the methylation patterns with microbial priming, and I've seen acetylation patterns linked with chemical priming. But of course, this is not exclusive. So, in terms of function, the modification to the histones impacts the structure of chromatin, thus influencing the accessibility of the genes, of course. And stress-induced histone modifications can lead to the activation or repression of stress-responsive proteins that allow the plant to dynamically adjust its gene expression patterns in response to the external environmental stimuli. So, in terms of the impact, the dynamic changes in histone modifications provide a rapid and flexible means for the plants to modulate their genetic responses under the variable stress conditions. Then we also talked about the infamous small RNA molecules, which is also attracting increasing attention. And these small RNA molecules have many, many different forms. A lot of research has been done on miRNAs, but also there is quite a lot of literature on long non-coding RNAs and many, many other different forms. So you also have the small interfering RNAs that is linked with gene knockout approaches, okay? And these are involved in any case in the so-called post-transcriptional gene regulation that actually alters the mRNA status, okay? This is very important for silencing techniques when carrying out gene silencing. And this is also essential when developing technologies like VIGs, virus-induced gene silencing, and so on. So this is RNA interference, the basis of RNA interference. So small RNAs can target specific mRNA molecules, thus leading to their degradation or their translational suppression. Okay? So changes in the abundance and activity of small RNAs under stress contribute to the precise fine-tuning of gene expression, thus allowing the plant to respond with specificity to different stressors. So the specific short small interfering RNAs in the case of RNAi will actually identify the corresponding transcript based on homology of the sequence, and it will actually bind to that, and with the use of specific proteins that make the so-called RISC module, they will actually start chopping the final mRNA, fragmenting it to small molecules. Exactly. So this will actually stop the translation of this transcript, and the short pieces that will actually occur will actually go back to restart the circle. Okay? So the regulatory role of small RNAs provides plants with a rapid and adaptable mechanism to modulate gene expression patterns, thus enhancing their ability to navigate the complexities of stress environments. And here you see a general map that shows the involvement of some of the key miRNAs that have been identified in response to abiotic stressors. So there is quite a lot of miRNAs that have been implicated in abiotic stress responses and have been actually functionally shown to indeed lead to healthier plants under stress when being manipulated at a genetic level. And in terms of the integrated impact, by the way, how long am I supposed to go until we have a break? Do you know the schedule? 1:15. Good, good. So in terms of the integrated impact, putting together everything, epigenetic changes in plants during stress conditions do not operate in isolation. Instead, they basically form a very intricate network where DNA methylation, the histone modifications I mentioned before, that could include again methylation patterns, and small RNA-mediated processes, crosstalk, and collaboratory influence gene expression levels. So this integrated impact basically ensures a very highly coordinated and very context-specific response to environmental stressors that allows the plants to basically fine-tune their gene expression profiles for optimal stress adaptation. And this is a diagrammatic representation of the way that epigenetic changes are basically remembered by plants. So in response to the stressor, the plant will actually will recover upon successful tolerance to the stress, but it will in the process develop a sort of epigenetic memory that will then be inherited to the future generation. And this is why I mentioned that seed priming appears to have this element, because we do see this intergenerational effect, because single treatment events at the seed level do show protection at a mature plant stage. We actually do see that. However, one comment I should make empirically is that although our seed priming technologies with chemicals work really nicely, and we do get protected plants that are even 30, 40 days old when we expose them to stress, we have seen that it's a little secret. If you do additional exogenous application on the plant as a spray or as a soil irrigation, in addition to initially treating the seed, we've actually seen even further improvements. So an additional priming event in a mature plant or a seedling following the initial seed treatment does appear to be even further improving the technology. We've seen that with a couple of our technologies, including the NOSH technology, and I think this is quite interesting, that it seems to be kind of like a booster shot for even further increasing the effectiveness of our priming technology. Okay. So, where was I? Yeah. So, in terms of the epigenetic crosstalk, the epigenetic crosstalk refers to the interdependence and the communication between different epigenetic mechanisms. We have lunch? Yeah. I'm actually a bit tired, so it comes really handy. Okay, slide 31. We stop here for lunch and we continue on this slide. It's okay?